And we're back. And as we hit this chapter, we finally heavily get into the foundations of psychopharmacology. Um, you'll see that concepts from earlier chapters come up here again, as do some humans that I'm a big fan of. The opening vignette here is not about so much a real story, but my good friend Santiago Ramon E. Cajal is here, and father of modern neuroscience. Yes. Um, I love it that the book says that he, you know, stood up to an outspoken and arrogant Italian named Camillo Golgi, and they had very different ideas about how the nervous system worked. Golgi thought that the nervous system was one big mush, one big net, all cells connected to all cells, not like plugged into, but continuously connected. Whereas Cajal pushed the neuron doctrine. At the bottom of the page, there's a link for the 1906 Nobel Prize, and I've looked that up for you. And yeah, just take a look at those two people and tell me who you would rather hang out with. I definitely prefer Cajal. I've read several books about him. I read his autobiography. I own this book, The Beautiful Brain, Drawings of Santiago Ramon y Cajal. And you should be able to name that neuron in that picture. The hint is it's like a coral that you find in the ocean, a fan coral, which is basically two-dimensional. So it's a Purkinje cell. And remember to capitalize that because that's a human name. And all these dendritic branches have spines on them, possibly 100,000 synapses. It's a stunning book, stunning. Cajal actually invented some protocols for photography, but most of his research was done, or his publications were done by drawings. This is another book I've read and I give to some people in my lab when they graduate. Advice for a Young Investigator. It's a really good book. And I've read this one. This is Cajal's autobiography. And here are some photos. I can't decide. I think this one is one of my favorites. Sitting in his lab with his microscopes. Notice, by the way, that these are all monocular microscopes. Modern research microscopes today are binocular. They were lucky to be able to get one of these tubes to do a good job, and to get two of them paired was a real trick. The resolving power of microscopes back in Cajal's day, pre-1900, the resolution was about as good as we have today with current regular light microscopes. But it was like looking through a straw. Only the center of the image was sharp, and the rest was not. So Cajal had to draw things. And there's the book. And okay, I can stop anytime I want. I love Cajal. I have so many Cajal stories. He was a hard worker, that dude. Okay, so that's page one. I'm not a fan of Golgi, and although his stain works really well, and I use it a lot, not for research, but believe it or not, for fun. There's the discussion of Otto Lowy in 1920, another Nobel Prize winner. like Cajal, he shared it. And there's Lowy. He's a nice guy. And his Nobel Prize was also shared with someone who will come up later in the chapter. I don't think I'll talk about it. But people like to talk about dreams, and dreams did play a role in Lowy figuring out the chemical nature of neurotransmission. Back in the day, there was a fight between what were sometimes referred to as sparks, who thought that neurotransmission was electricity going between cells, and squirts, who thought that chemicals were moved from cell to cell. And arguably the sparks made more sense, but this isn't philosophy, this is science. And Lowy had a dream, and he figured out a way to test a theory about chemical neurotransmission, and the squirts largely won, and it's described over here. Parasympathetic, we've already seen that term before, and the nerve involved was the vagus, which is related to the word vagabond for wanderer. And the vagus nerve, when stimulated, slows the heart, and it does so with acetylcholine, cholinergic. Henry Dale is the other Nobel Prize winner, and he's appropriately famous, and I don't dislike him if you care. They both did really good research. So synapse, Sherrington, another Nobel Prize winner, and the point of functional contact between a neuron and another cell, usually another neuron, but as the book points out, it can be a muscle or a gland. These terms are important. I use them so frequently I forget that they're novel to you, but the presynaptic cell is often represented as the end of the axon, the swelling at the end of the axon, although the swellings may be along the ends of the axon. That's the presynaptic cell. That's where the neurotransmitter comes from, typically, and it moves across the synaptic cleft, which will be discussed a little bit below, to the postsynaptic cell. So you have a presynaptic cell, and of course the presynaptic cell owns the presynaptic membrane, and the postsynaptic cell owns the postsynaptic membrane, and by the end of this chapter you will know that. The electron microscope, if it weren't for that, we'd probably still be not sure that Cajal was right about the neuron doctrine, but because light microscopes, as I said, back around 1900, their resolving power was as good as it is today. And the reason for that is not because of the optics of the glass, it's because of the nature of light. And light is electromagnetic, and it has wave-like properties. And when you're trying to look at objects with waves, if the waves are bigger than the object, it's very hard to see. So we have now supplemented light microscopes with electron microscopes that use electromagnetic energy with higher frequency, and we can actually see the synaptic cleft. And you will see pictures of those, I think, on the next page. These terms at first may seem confusing, but they're not. Axodendritic means from an axon to a dendrite. Axosomatic is axon to soma. There's even axoaxonic. So again, none of these names are meant to confuse you. Pyramidal neurons, we mentioned those before already. There's the synaptic cleft or gap. Most graphic representations of pre- and postsynaptic membranes and the synaptic cleft exaggerate how big this cleft is. It is not very big, and we'll see that thanks to electron microscopes in a second. I really like that this book mentions the postsynaptic density. When you see electron micrographs of tissue, there's two major kinds of electron micrographs. One is called a transmission electron micrograph. Transmission EM, electron micrograph, or electron microscopes, they send electron beams right through the tissue. The tissue has to be sliced very, very, very thinly. And to do that, we embed the tissue in plastic, believe it or not, and we cut it with glass knives. It's back in the day, it required a great deal of skill. The science of neuroscience and pharmacology largely is based on transmission EM. But you've probably seen a lot of pictures of scanning EM where you see pictures of bug eyes and other things where it looks three-dimensional and there are shadows. That's electron microscope too, but they have covered the thing they want to look at, often with like powdered gold. So electron beams bounce off of it instead of going through it, and they capture them and reconstruct the object that they bounced off. So in this book, I'm sure we see both, but I think this chapter started off with scanning EM, the picture that started the chapter, but we're about to see transmission on the next section. The postsynaptic density is very important. This idea that there's this big, crevasse between the pre and postsynaptic membranes is not true. They are very close together, and there are also little fibers connecting them. And by fibers, I don't mean hairs, but processes. And you've seen in the videos, if you've watched them, how cell membranes often have things growing out of them, and things can grab to things, and think about that. And synaptic vesicles, they're like little cells within cells. They have bilipid membranes, and they have stuff inside, and they float in an ocean that's not outside the cell, but it's outside the vesicle. And they're their own little worlds, and it's complicated. There we go. So there's a very nice transmission EM. It's not colorized. Here's the functional synapse. Notice you have the presynaptic membrane and the postsynaptic membrane. And if we zoomed in, we could see that they are bilayers. And actually, you can see the bilayer over here. These are synaptic vesicles, and they come in different sizes and shapes, and that can be significant. This is a mitochondrion, energy source for the cell, which occur all over the place. But notice the postsynaptic cell, which looks kind of boring. The dendritic spine in this case. Notice you can see a thickening on the inside of that cell membrane, and you can see a thickening between the pre and postsynaptic membranes. That's all very real. Now on the right is magic. This didn't exist when I was you guys. So these are spines on a pyramidal neuron, and you know what they look like? They look like pyramids, and it's human. And they got a fluorescent dye into the cell, and they used a confocal, which means you've got things coming from two directions, and you can get a much better three-dimensional view of things. And all of these are spines. And look at the thin necks, and then they swell, and they're long and they're short. And those puppies are changing size and numbers by the second. That is extremely alive. And now we have a picture of exodendritic, exosomatic, and exoaxonic synapses. And we've already dealt with the words. They're self-explanatory. Presynaptic. So before the synapse, presynaptic. That's the cell that's talking to the second cell. Presynaptic inhibition often involves the reduction in neurotransmitter release sort of like putting a gag over your mouth. It's harder to release the neurotransmitter, but you can also change things to cause presynaptic facilitation. If you can imagine it, it's probably true. And as I said, synapses usually occur between one neuron and another, but obviously your muscles move because neurotransmitters are released out of them at the neuromuscular junction, which isn't overly important in this part of the chapter, but it's a good thing to know. Oh my. Neurotransmitter synthesis, release, and activation. 1, 2, 3, 4, 5, 6. I remember being in grad school, drilling myself on those 6 mechanisms when I took psychopharmacology then. The good news for you guys is it's an open book test. This is one of the questions that you'll be responsible for being able to answer, but there you go. What should be true if something is a classic neurotransmitter? So there we go. There are kinds of neurotransmitters, and not to overstate that I'm wicked old, but when I was taking this course, I think we had 6 or 7 neurotransmitters. Oh my, we have way more than 6 or 7 now. So here's the table, table 3.1. It almost certainly, yes, it is. It's one of the questions in the back. So you have classical neurotransmitters on the left. Those are of the type that were true back in my day. And non-classical neurotransmitters, some of those were being just discovered as I was getting out of graduate school. So you have amino acids, and boy, are they important. And glutamate and GABA and glycine. GABA, we're going to spend a fair amount of time on for sure. The monoamines, and you probably can guess that they are monoamines because they have one amine group. Dopamines, norepinephrine, serotonin, and I thought I turned off Facebook. They are really important. Serotonin is important for, at least historically, has been important for treating depression. And norepinephrine and dopamine are very important for a number of conditions. For example, Parkinson's and schizophrenia, and serotonin has been important for depression. Acetylcholine, we've already run into that one, cholinergic. That's Lowy, and he called it vagus stuff because it came out of the vagus nerve, and that's the first neurotransmitter characterized in the body. And purines, those purines, they weren't part of my life. And then over on the right side, neuropeptides, endorphins and enkephalins, those are opioids. And I spent a lot of time dealing with them. And our old friend naloxone, which is Narcan, that's a receptor antagonist for some receptors of those. Corticotropin releasing factor, orexin, hypocretin, many others, and anandamide. That deals with your brain's own marijuana systems. Gases, Nobel Prize for research relating to nitric oxide will be discussed in this chapter. The Viagra keeps coming up in the chapter, and that is related to nitric oxide. But unbelievably, carbon monoxide is a gaseous neurotransmitter. Hydrogen sulfide. It's mind-bending how complicated it is in there. Okay. Um, Up here, neuropeptides, endorphins, and enkephalins. Again, those are opioids. There we go. But the book, unfortunately, has gone modern and says abused opioid drugs. I don't. You can speak as the book speaks, but I would much prefer that you think of drugs of abuse as being outside the body, and therefore I prefer to call them opiates, IATES, and not opioids, which are what your brain makes. It's not important. It's just I have no clue why the world changed between when I was very active in that research field and now. Box three. Box three is giant. It goes on for the next flip and maybe the flip after that. I can't expand on it. It's important. It deals with a lot of topics. Well, mostly it focuses on sleep. Um, by the way, hypnagogic hallucinations are the kinds of hallucinations you get as you go to sleep, and thus the term hypnagogic. Hypnopompic hallucinations, which I don't think show up in this chapter, are those that you have as you awaken. Sleep paralysis. I'm a descending pain inhibition guy. There are descending brain to spinal cord pain inhibitory mechanisms, and I showed you, the reference from long ago about opioid and non-opioid uh, pain inhibition systems, anandamide, that activates endocannabinoids. That's one of them. They go down into the spinal cord and can block pain, basically at the first synapse in the nervous system. Sleep paralysis has parallels in that the brain has descending brain to spinal cord motor inhibitory systems, where your motor neurons can be inhibited by these cells, which is good because if the motor neurons were not inhibited, you would literally act out your dreams, which would probably disrupt your sleep and disturb people around you. Sleepwalking, by the way, is not typically a dream. So there's orexin arrows all over the place and neurotransmitters all over the place. Obviously, the authors are very into this. The RAFA nuclei are very important to me. 5-HT is an abbreviation for serotonin. RAFA means seam. So you have left and right eyes, left and right nostrils, left and right hands. There is no left and right RAFA nucleus. The locus ceruleus came up in the last chapter. I mentioned a color, cerulean blue. That's a primary source of norepinephrine in the brain. Notice it's affected by orexin. There is a left and right locus ceruleus. The ventral tegmental area I mentioned in the last chapter. When you think about reward and addiction, that has to come up. And there's left and right of that too. So everything up here is left and right except for RAFA. And some of the RAFA are very, very important for descending opioid pain inhibition. And loss of orexin neurons, and we have animal research and human research. It's very elegant how they work this out, very elegant. And narcolepsy, it can be extremely debilitating. It can be as bad as epilepsy. You can't drive, you can't carry your child down the steps because you may have a narcoleptic attack. It's not a joke. And it's been used in jokes, unfortunately. Okay, so now there's a mention of gaseous neurotransmitters, and there's nitric oxide, and I mentioned that already. We've mentioned over and over bilipid membranes, and things have to be, able to deal with, the bilipid layers. or else they can't get into cells. Boy, nitric oxide doesn't care. The nitric oxide, once it's produced, it just goes everywhere. It's like throwing a pebble in a pond. It just ripples out and it goes through the cell membranes of the cell that produced it. It can go to the cell preceding that cell. For example, here we have a purple cell that's talking to a blue cell. If the blue cell produced nitric oxide, it could pound right back into this cell. And by the way, this area is surrounded by blood vessels, and the nitric oxide can go to them and cause vasodilation. Because when your brain becomes more neurologically active, it needs to become more metabolically active. It needs more glucose and oxygen, and it has basically no stores. So if you want your brain to be able to keep up with itself, you have to have the blood vessels adjust to the brain's needs. And nitric oxide is critical for that. Neuropeptides are synthesized by different mechanisms than classic neurotransmitters. That's discussed very nicely in the book. Notice these little tendrils hanging down here, microtubules, and you saw the picture of the little guy carrying the vesicle along the microtubules. That's part of what goes on there. This is discussing large and small vesicles. As I said, they can have sizes, and the peptide neurotransmitters are schlepped down the axon in the large vesicles as part of larger proteins, and these subparts of the protein are snapped apart to produce the peptide neurotransmitter. It's confusing, but you'll get it. Notice that some of these vesicles have mixed contents and some of them don't. We used to believe in over here, there's Dale's Law. Dale shared the Nobel Prize with Lowy. He didn't say it, but people used to think he said that each neuron releases only one neurotransmitter. He didn't, and they don't. It's way more complicated. You have postsynaptic receptors, and you see shape differences here. Some of these receptors, the green ones, are channels, and some of them, also transmembrane, are not channels. And ionotropic and metabotropic will become common terms in a bit. And by the way, the postsynaptic membrane, again, can produce something like nitric oxide, which can go pounding back into the cell and affect this cell's release of future neurotransmitters. It's a dialogue in there, yin and yang. Okay. So this is a discussion of neurotransmission using neuropeptides, and there's some details about what goes on in the neuron here. And they mentioned rough endoplasmic reticulum, which probably you then think, well, if there's rough, there must be smooth, and there is, but not important to this discussion. And things are budding off and going into the Golgi apparatus, and then the big vesicles are produced, and the little guys walk them down to the to the axon terminal, where, of course, here it looks like we have this giant synaptic cleft, but we don't. Okay, neuromodulators. That's, as the book points out, this is a bit vague, but it's an important concept. The concept being that things don't always work like classic neurotransmitters. You can have neurotransmitters, which some may call neuromodulators, that don't directly do things, but they modulate how well other things happen. And that's a nice discussion there. Classic neurotransmitter involves exo, mean exit, exocytosis out of the cytoplasm, and recycling of synaptic vesicles. So here we have exocytosis. It says it's a remarkable process, sure, unlike everything else. And here's a picture that gives you an idea of what's going on. Now, let me caution you on this picture. Notice, number 2 here. Where's number 1? There we go. Number 1's over here. But number 2 is an action potential hits here. This transport system here going on between the soma and the terminal, and remember it's also bidirectional, that's going on independent of the action potential. So these vesicles are coming down here and things are being processed and stored, and they dock independent of the action potential. But when the action potential hits, remember the cell is minus 70, and it says depolarization, which means the action potential in this case opens calcium channels. Now, I encourage you to go back and look at the relative concentrations of different ions inside and outside the cell. And as I mentioned when we were looking at that table, there's a pot full of calcium outside the cell, not too much inside. When those channels open, calcium is going to go through those channels into the cell for 2 reasons. One is differential concentration. There's a ton more calcium outside than in, and also charges. Calcium is positive, and the negative charge inside the cell is going to suck it in. And when that calcium comes charging in through that channel, it's going to trigger a cascade of events. And the book uses this term the same way I do. When you say a cascade of events, what you're saying is it's complicated and I don't really understand it. It causes a cascade of events that causes these vesicles, which isn't really shown here, which are usually already docked where they're needed. And remember the transmission electron microscope picture where you have that thickening where there's a functional synapse. Well, on the presynaptic side of that thickening, you're going to have vesicles already docked, just like a harbor, ready to go. And when the calcium hits, they form what are called omega bodies, because if you look at Greek symbols and all, that sort of looks like an omega. It opens the pore. The vesicle does not leave, it just opens, and the neurotransmitter can move out into the synaptic cleft, which, as we know, is much smaller than that. And then the vesicle is brought back and recycled, and there's different mechanisms, and the book will talk about that in the next pages. And now these pictures here are all ion channels, and they can have excitatory effects, they can have inhibitory effects, and the second cell may or may not produce an action potential. Excitation and inhibition are equally important in terms of information exchange in the nervous system. We just had the 4th of July, and up at the top of the Washington Monument, there are 2 lights, one if by land and 2 if by sea. And you could light those lights, or you could have them lit all the time and turn them off. And if I turn off 1, they're coming by land, and if I turn off 2, they're coming by sea. It's equally informative. Active zones, we've seen the active zones. Okay. Life cycle of synaptic vesicle. This is much more realistic. Notice it has docking points and specialized proteins, and you've seen the videos, man. They really exist, and some of them are transmembrane, and they kind of kissy here, and they're ready to go. And when the calcium hits, they pop open. There's the calcium, and the neurotransmitter can be released. And the book now goes into details that I will not repeat, but they're probably in the chapters, the chapter questions. I forget. Botulinum toxin, one of the most toxic substances that we know of. Never mess with botulism. You can lick a fork with just drips of contaminated liquid with botulinum toxin on it, and you can die. You do not mess with it. So there's a nice discussion of that there. Whew, look at that, baby. So on the left, it's a little hard to see when you first look at it, but that's cut in two, sort of like a sagittal section. Notice it has a lipid bilayer, as does the cell. Notice that there are many elements that are transmembrane and some that are not transmembrane. On the right, it's more calming. It's the outside, and look at those things sticking off. Just waiting for it to stick to. And you see the video. These are real. And this is glutamate, which is a classic neurotransmitter, and they're using it as an example of how it can work. So we have different models of what's going on here, and we haven't completely figured out. So they talk about three mechanisms: clathrin-mediated endocytosis, ultra-fast endocytosis, and kiss and run. I just love that term. So I usually talk about kiss and run on the next page. There are pictures of those possibilities. And in all cases, you do not throw the vesicle. It's not like you open a car window and throw a water balloon out on the street. It's a bit more like you're inside your house with a screen door and you throw a water balloon at the screen. The balloon, the vesicle, stays in the house, but the contents egress. And then there's a fourth mechanism, bulk endocytosis, and exocytosis, leaving the cell, endocytosis, coming into the cell. And here we go. Lipid and gaseous neurotransmitters are not released from vesicles. Why not? The vesicles can't hold them. They don't see, the membranes just don't exist. They're like ghosts. They go through walls. Retrograde messengers, the axon. I haven't seen the term in the book. I could search, but let me just say that if things are moving from the cell body to the terminal, that's called anterograde transport. From the cell body to the terminal, anterograde. I said it was bidirectional, though. If things are moving from the terminal to the cell body, that's retrograde. And these are retrograde messengers. So, you're waiting for a signal from another cell, and the cell sends you a signal by exocytosis and classic neurotransmitters and ions or not ions, as we'll shortly see, and you get the message. But one of the things you might do in response to the message is produce a gaseous neurotransmitter, pebble in water, that just blows everywhere. And part of everywhere is where the message came from. So it can indicate to the presynaptic cell, message received. It's like, I got it. You signal back. Okay, there's differences between classical and gaseous and lipids. Notice this orange cloud here. That's what I was talking about. That's the ripple. And one thing they're not showing you here is that within this cloud, there are probably capillaries. And one of the reasons that you may provide more blood to these cells is because the gaseous neurotransmitter hits those capillaries, they dilate, and everybody's happier. Autoreceptors. Older people who have trouble hearing. How do they speak sometimes? They often almost yell. Why do they almost yell? Their autoreceptors, their self-receptors, their ears aren't working very well. So we modulate our volume because we can hear ourselves. And when we get a good signal back, we know we're talking loud enough, and we titrate. We talk just loud enough to get the message across. People with hearing loss they can't hear themselves talking at normal volume. So their automatic systems upregulate their volume and they talk louder. And to some extent, that's what can happen, certainly with terminal autoreceptors and even somatodendritic autoreceptors. And there are pictures on the next page. So there's pictures. Here's the terminal autoreceptor. And you really can think of that as being very much like your ear. So if you hear excess neurotransmitter coming back to there, you say, I got it, and you can turn down the volume a bit and reduce neurotransmitter release. Over here, although not all neurotransmitters are released onto the soma, but this can inhibit the willingness of that cell to generate additional action potentials. So kind of similar. Transporters and reuptake. Reuptake is one of the major ways we turn off neurotransmitters. We went to all the trouble of making them. We release them into the synaptic cleft. They talk to the postsynaptic receptor site. That's a dynamic interaction. They attach and they are released. And they can attach again, but there are mechanisms in the presynaptic cell that will grab these neurotransmitters that you just released and take them back. And of course, once taken back, they're typically stored again in vesicles and they are reused. And acetylcholine, in contrast, is often destroyed in the synaptic cleft. It's a fast on, fast off neurotransmitter. If your fingers were run by neurotransmitters that were slow on onset and slow on offset, you couldn't play a piano. You couldn't do anything fancy with your fingers. So it's kind of expensive, acetylcholine at the neuromuscular junction, but it's for good cause. But other neurotransmitters, many of which that we know and love, and here's dopamine, norepinephrine, and serotonin, they're often turned off by reuptake. And some of the therapeutic drugs that we use to treat things like depression are messing with reuptake. Cocaine blocks the transport for dopamine, serotonin, norepinephrine. A graduate of the University of Scranton was a discoverer of the cocaine transport mechanism. And there's an overview of, glial cells. I forgot to mention that. The reason I didn't mention it was that when I was you, they weren't important, but now they are vitally important, and particularly for some excitatory neurotransmitters. The glial cells take up excess glutamate, for example, and glutamate is such a powerful excitatory neurotransmitter, it can literally excite this postsynaptic cell to death. And one of the ways we get glutamate out of there is we sequester it in glial cells, and they can give it back later, but the more important thing is it gets out of the way of this cleft. When you get a head injury, your bell rung, a concussion, these glial cells don't do a great job of soaking up the excess glutamate. And some people are put into pharmacologically induced comas to calm down the nervous system so they don't have seizures until the brain and the glial cells can get their act together and this system goes back to normal. So it's important. Enzymes, right, transporter right into the axon terminal, transporter into the glial cells. It depends on the neurotransmitter, where the transporters are. Second messengers. They're complex. And I think somewhere in here, the book says it's too complex to cover in the book. The good news is you have an open book, and the questions that are given can be easily answered from the book. So down here is the comparison of ionotropic and metabotropic receptors. Ionotropic ions, ion channels, and we've already alluded to those. And I believe I mentioned that they often have 4 or 5 subunits. Now, what are subunits? You saw in the video that there are these tall structures that can sometimes gather together. And if those tall structures are transmembrane proteins, and you put 4 of them or 5 of them in a circle, you create a channel or a pore. And they're very important. They're very fast because the neurotransmitter hits the receptor part of that complex, and those proteins can change shape really fast. That ion channel can snap open or snap close. Ions can flow or stop flowing, and things happen quickly. So they contain an intrinsic ion channel. Well, they don't contain a channel. They produce a channel. I would rather use the term here receptor complex. And I mentioned Star City a lecture or two ago. So these things are big, and their assembly produces a channel, and they can sometimes have multiple receptor sites on them too. They tend to not be coupled with second messengers, and they are fast. They are wicked, wicked fast. Metabotropic receptors, they're transmembrane, very transmembrane. They do not directly open and close ion channels. They are not fast. They activate often G proteins in response to, which doesn't make sense now, but will in a page or two. Are they coupled with second messengers? Yes, they are much more complicated. They can even affect DNA expression, which and a lot of what psychology is interested in, particularly if you're trying to treat things like depression and schizophrenia, those things, or anxiety and fear, those things last a long time. They are not zip zip like playing the piano, finger down, finger up. So metabotropic receptors are really, really important to psychopharmacology. A lot of the drugs we deal with those receptor complexes. They are coupled with second messengers, and they are not fast. And their effects are, slower and thus not fast, but they can also affect DNA. And by affecting DNA, they can affect DNA, RNA synthesis, and you mess with protein synthesis, you're messing with the structure of the cell. So they're really important to what we do here. Okay. So on the left, there are your cluster of, in this case, 5 transmembrane protein complexes. Notice that there are little dimples on this one and this one. In a later chapter, you will find that that is almost certainly an acetylcholine receptor. And for this complex to fire off, which means for the ion channel to open, both of these sites have to be occupied by acetylcholine. And then this channel, which is now closed, will snap open, and sodium can come in. And boy, this is also a brilliant picture of the bilipid membrane with the hydrophilic, and hydrophobic sections. So here's a graph over here, and they have shown two neurotransmitter molecules docking again. It's acetylcholine. So those are ligand. There were channels earlier, and we had ligand gated channels. Ligands are chemical neurotransmitters often, and the ligand, in this case acetylcholine, is going to dock, and when two of these sites are docked, they facilitate each other. This thing pops open, and off we go. Not overly important for psychopharmacology, but remember there are also voltage-dependent channels, which are not going to be discussed here. Desensitization. This can be problematic when you're using some of this stuff therapeutically, because this is dynamic. These are living, each of these are living proteins, and you hit them, and there's a dynamic interaction between the green ball and the purple transmembrane structure, and the receptor complex, the receptor itself, which is part of the receptor complex, kicks that ball off, and then the thing will close and can reopen, but you overdrive this, and things can change. It can become less responsive, for example, from the overdriving, and we can show this. Okay, now here we're talking about calcium as a second messenger. When we deal with the nitty-gritty of neurotransmitter receptor sites, metabotropic receptor sites, calcium isn't going to be vitally important there. But remember, in the picture of the action potential and how it causes exocytosis, there were calcium channels, which are ion channels, which are like these channels, that are not ligand sensitive, they are voltage sensitive. And when those open and calcium enters the cell membrane, it talks to the inside and can act as a second messenger. NMDA receptor. We're going to spend a lot of quality time with the NMDA receptor in block 2. Still it will be end of block 2. This is really important because it's related to glutamate, and glutamate is going to be very important to this course. Chloride. I skipped over it a bit earlier. It talks about the chloride entering the cell and hyperpolarizing it. That's often what's described. I don't fully agree with that. It's more like an antacid or a buffer. It kind of locks the cell membrane up at the resting potential. It doesn't so much cause hyperpolarization, but it has the same effect, because if you're locked at the resting potential, you can't get to the threshold of excitation. So chloride's important. And GABA, we're going to spend a lot of time with GABA. Again, notice this chapter 8. And here we go, metabotropic. And as I've already mentioned, they don't possess a channel or a pore. And there we go. So there's our bilipid membrane again. And look at this puppy. Notice it's like a string of fireworks. Section 1 is connected to section 2, 3, 4, 5, 6, and it's got a little squiggle outside. The binding site tends not to be there, but the G protein binding site is down here. So the G protein is often docked there. And in this picture, there's the neurotransmitter hitting the receptor Now, in this, it looks like the receptor is the receptor, but it's not the receptor. In ionotropic receptor complexes, we had 4 or 5 subunits. This has 6. Oh no, 7. So somewhere in this mess, possibly multiple times, you have receptors for these neurotransmitters, which can kick your G protein free, which can float over here and tickle the insides of this ion channel. This is a G protein gated ion channel, and it's an ion channel. And this ion channel is going to be 4 or 5 subunits, just like we saw over there. And this is more complicated. The G protein is kicked loose, and it talks to this pink thing, which is not transmembrane, but it's close. And it can release second messengers into the second cell, and they can do a million things. So we have G-coupled receptors, potassium. Now it says here, when the potassium channel is open, potassium ions flow out of the cell and the membrane is hyperpolarized. That I do not disagree with. Remember, at the end of the action potential, the cell gets more negative. It hyperpolarizes. That's because potassium channels are open and potassium gets to go and the cell becomes more negative. So this is important. So you open these potassium channels, the cell will hyperpolarize, get further away from the resting potential, and it's harder to excite the cell. And look at who uses it. It's a common mechanism of synaptic inhibition used by acetylcholine, dopamine, norepinephrine, serotonin, and GABA, including some endorphins, which are opioids. Dude, that's a lot. That's an important thing. There's the effector enzymes, which were in that graphic. Oh, here we go. This sequence of events is an example of a biological cascade. And they've sort of explained it, but I still think that means there's more, and we should not talk about it. That's certainly how I use it. Allosteric modulators, here we are down there. Notice they go to this like Pac-Man version of receptors. The receptor site is not a receptor. It's a receptor complex. It's multiple proteins or multiple segments of a protein. And it can have docking sites for many things, as we will see in later chapters. So here's the agonist alone. Here's a modulator that increases the effect of the neurotransmitter. Here's a modulator that decreases the effect of the neurotransmitter. And here's a modulator that stands alone. The cheese stands alone, and nothing happens. Here we go. Finally, we admit defeat. Second messengers work by activating specific protein kinases within the cell. Second messenger mechanisms are too complicated to completely be covered in this text. Amen. You are responsible for being able to answer questions, and they're back there regarding second messenger mechanisms. There are pages of this stuff. It is there. That's a better graphic. So now we see what the pink thing releases, the messengers and the protein kinases and substrate proteins and cellular effects, which can include inside the nucleus. It is wicked complicated. And there's a lot of terms that are important. Second messenger systems and protein kinases. Oh my God, it's an hour. I have been running on. Different protein kinases for different second messengers. And up in here, we'll see that, there's a word I love, plethora. There's a plethora of phosphodiesterases. And it's complicated, but that means we can design drugs that specifically target one target like PDE5, Viagra and Cialis. So it's useful. It's pharmacologically important. Certainly, I wouldn't spend a lot of time trying to memorize it, but be able to explain it because it is at the end of the chapter. TRK receptors, TRK receptors, worth knowing. Transmembrane, not a channel, but when neurotrophic factors affect the outside, you get changes inside, activation of other proteins. It's complicated. It's a cascade. Drugs can either increase or decrease the rate of neurotransmitter synthesis. Amen. Parkinson's, which is a bad one, and I've mentioned I've watched someone die of that. L-DOPA is a current very popular treatment for Parkinson's. When it works, it is literally magical. There are videos where people go from being almost immobile to being quite normal on L-DOPA. Unfortunately, it doesn't. It's a symptomatic treatment. It doesn't stop the disease progression. Parachlorophenolamine I highlighted because that was my first research drug before I got into naloxone. PCPA, they don't say PCPA, but it is PCPA. Parachlorophenolamine inhibits serotonin synthesizing enzyme tryptophan hydroxylase, and that really disrupts serotonin levels. Physostigmine blocks the enzyme acetylcholinesterase. That's how you turn off acetylcholine. And I'll tell you, man, if you release acetylcholine and you can't turn it off, you get spastic paralysis and death real fast. You don't mess with physostigmine. There's an overview of everywhere drugs can affect neurotransmission. By this point in the chapter, it shouldn't be overly frightening. Go through that step by step and get a grip on what those points are and how they work. A lot of this is review of what we've already gone over. Dendritic spines. We saw that the picture of the scanning confocal at the beginning. And this is an interesting parallel with autism, Alzheimer's, and Huntington's, and normal. You don't want too many spines. You don't want too few spines. It's a very interesting overview. Endocrine. A lot of glands, a lot of things to read here. Adrenal medulla, adrenal cortex, open book test. These are important. Our friends, the glands, they do lots of things. And if you know anybody with diabetes, insulin is important. And the islets of Langerhans are important. And where hormones come from, sex hormones. It's just a gentle overview because psychopharmacology deals with some of this. The pineal gland, I have a little cyst on my pineal gland. It releases melatonin, important for daily rhythms. The pineal gland in birds is right up under the skull. It can directly respond to light. Pituitary, I've already referred to. Master gland, million hormones. And here we go with the structure of it, and anterior and posterior. vasopressin and oxytocin. We're going to see those a million times later. There's a great figure here that I believe there's a study question that taps into this fairly heavily. There's the anterior pituitary and the posterior pituitary. They share the pituitary stalk, but notice that vasopressin and oxytocin on the right, those axons are going right down into the posterior pituitary. But the axons that are relevant to the anterior pituitary are talking to the blood vessels that feed to the anterior pituitary. Not life and death differences from our perspective, but it's true. So you have to know that and what those hormones do for a living, and that's a study question. And we've seen this structure before, and they love this. And here we go. And I keep threatening you with things that go into your nucleus and affect your DNA. Well, there are some hormones that do exactly that. And you mess with what parts of DNA are active or inactive, and DNA RNA protein synthesis, it's huge. It's very huge. There's a nice discussion here on the right of D2 receptors, dopamine receptors, which we often target traditionally when teaching, when treating schizophrenia. There's a discussion here of them being related to prolactin, and it's nice. It's a good discussion. Why didn't I make prolactin? Anyway, yes, prolactin. There it is right there. And here's a picture of that relationship. So if you block 50% or more of the D2 receptors, prolactin levels just dump. But if you have 50% or more available, your prolactin levels are pretty okay. This is a big discussion that I can't improve on. The lab I was in was very interested in sex differences at the end in relation to pain inhibition. And this deals with sex differences in drugs of abuse. It's complicated. And almost all researchers historically have used male mice and rats. And why do we do that? It's not that we hate females. It's that they're more complicated. And you've got cycles, and the cycles are coming right up on the next page. And the timing of when males and females or females are given drugs can have a dramatic impact on the impact of that drug on that person or animal. And this is an important discussion. It's a mature discussion. And more research should be done on females in terms of drugs. But to do that research, you need to pound more money because you can't just grab a male. Yes, you can grab a male, but females, they have stages, and you have to study them in all those stages. So more animals need to be looked at, or the same animal across time. It's way more complicated. And I think we are out of here. I will spare you the reading of these since I've gone over an hour, and I usually like to stop. So, somewhere early in the chapter, and you'll see it linked with this discussion, there's an animation of how the postsynaptic membrane is organized. The book doesn't give a link to it, but I found it, and I posted it, and it's pretty. And it gives you, like the video that I encourage you to look at the other time, it gives you a real feeling for how synapses are are organized. Have a nice day. We are done with whatever chapter this is. It's not important, but we're done. See you next time.